Welcome to Unacademy. I'm Farida and I teach biochemistry for NEET PG. So, as we say, let's crack NEET PG. I've done my graduation, post-graduation and MPhil in biotechnology. I'm pursuing my PhD in biotechnology. I've qualified MP Set examination and I have a teaching experience of 8 years in biochemistry, microbiology and molecular biology. Today we are going to discuss the structure and organization of DNA. Before commencing the topic, let us have a little introduction for Unacademy. So these are the educators for NEET PG at Unacademy. They are the top educators all over India. Then there are special classes where you can ask your one-to-one questions directly with your educators. There are free test series, free mock tests and weekly tests, which as I always say is very necessary for qualifying exam to understand your progress, how much you have understood. To prepare for the examination with Unacademy, it is very easy because there is flexibility in timing. The language you can choose as per your comfort level and there are unlimited classes. If you have seen one class once, you can see it over and over again. It's almost like a classroom-like feel because you can directly chat with your educator. There are question and answer session and these sessions and these classes you can take anywhere, anytime. It is quite easy to install the app. Just go to Play Store and type Unacademy Learning App and you can easily install the app. Then for Plus subscription, there is a minimum subscription amount. By paying that amount, you will be eligible to access all of the lectures which are available on the Plus subscription. There you can have direct chat with your educators, various courses like crash course, short-term courses for various upcoming exams. They can keep on conducting courses every now and then. So you can easily go and get your subscription and start learning. For this subscription, you have to pay a very minimum amount, that is around, it is different for different packages. There are packages for 1 month, 3 months, 6 months, 12 months and 24 months. It is my personal suggestion that go for a 12-month and 24-month subscription so that you can make the most out of the time and the educational videos which are available. Then again, for getting more discount, you can use my referral code, that is FBO1, and you will be getting 10% extra discount by using my referral code. So just don't forget to get your extra 10% discount. Remember my referral code, that is FBO1. So let's begin with our topic, that is the structure and organization of DNA. The structure of DNA was proposed by Watson, Crick and Wilkins. These were the people who proposed the model of DNA, how the DNA would look, how the DNA is actually organized in the body. Then this structure which was given by Watson and Crick, let us have a look of special features of these. It is right-handed. It is wound in a right-handed double helical manner. Then there is a rule for pairing adenine. In last lecture, we saw about the chemistry and metabolism of nucleic acids. In that, we have clearly seen what are adenine, what are the bases. So, in DNA, adenine pairs with thymine and guanine pairs with cytosine, right? And they are bonded with each other with the help of hydrogen bond. So these hydrogen bonds, they are present in doublet between A and T, and they are present in the form of triplet in case of G and C. So adenine and thymine, they are bonded by double hydrogen bonds, and guanine and cytosine, they are bound by triple hydrogen bonds. So as we have seen that it is double helical in nature. This double helical is anti-parallel. Anti-parallel means one of the strands runs from 5' to 3' and the another strand which is opposite to it runs from 3' to 5'. That means they are anti-parallel. About hydrogen bonding, we saw that the hydrogen bond is present between the bases in the structure of DNA. Here you can see that A is paired with T. Here again T is paired with A, C is paired with G. So this is the pairing of bases. And how these two strands, if we just straighten these strands for a while, so how it will look like? It will look like something like a ladder. And these ladder, the steps are formed by bonds between bases. And this is the polynucleotide backbone. Okay. As you can see that it is right-handed helical. So when a right-handed helical is formed in DNA, two types of grooves are formed. First one is minor groove and the second one, the deeper one, is major groove. So as the name suggests, minor groove and major groove. So there must be some uses of these minor and major grooves. So what are the uses of these minor and major grooves? These are the places where various proteins, they come and bind and they regulate the gene expression. So these are the special sites for binding of various proteins which regulate the activity of genetic material. Major groove and minor groove. Another thing that we should see in this structure is for a complete turn, which if for example it starts from here, it will go like this, and like this. So this is a complete one turn, a complete circle it is forming in a spring form. So this complete circle, it spans how much long it is? It is 34 angstrom long. And what is the thickness of this genetic material? It is 20 angstrom thick. So you should remember that the length of a complete turn is 34 angstrom and the width of the genetic material is 20 angstrom. Then in a complete turn, in this complete turn, how many nucleotides are there? So in a complete turn, there are 10 nucleotides pair, 10 nucleotide pairs. Okay, 10 nucleotide base pairs. 10 nucleotide base pairs. So for each base pair, how much rise would occur? It is very easy to calculate. Just 34 by 10. So 3.4 is the answer. By each base pair, the turn increases by 3.4 Armstrong. So the height of the whole helix becomes 34 Armstrong and the thickness is 20 Armstrong. Just don't forget these two figures because many a times questions are asked on this. They'll be giving you the number for the thickness and height of the helix and they'll be asking it is which form of DNA. It is B, it is C, it is D. So if you will remember these figures, you will be able to identify the B form, which is the most prevalent one that we'll be seeing in our next slide. Then another important thing is Chargaff's rule. That means the number of purines are equal to number of pyrimidines. Purines are always equal to pyrimidines. Okay, so it means G, A, these are the purines, their number would be equal to the pyrimidine in DNA. Today we are seeing everything about DNA. So the amount of purine will be equal to thymine and cytosine. Chargaff's rule says the amount of purine will be equal to amount of pyrimidine, and it is but obvious also because pyrimidine pairs with purine and for A it pairs with T. By the help of hydrogen bonds, G is paired to C. With the help of triple bond, A is bonded to T in the form of double bond. Then there are different types of DNA. And what are these different types of DNA? A, B, C, D and E. These are the right-handed ones. And Z is the left-handed. Right-handed and left-handed. So all of them are right-handed. Those who are falling into a same series, they all are right-handed, and the one which is out of the series is left-handed. So it will be very easy for you to remember that which one is right-handed and which one is left-handed. The one which is out of the series is left-handed, and the one which is and all those which are in the same series are right-handed. So you will be able to remember right-handed and left-handed. So left-handed, only Z is left-handed one which is out of the series, and otherwise all are right-handed. Now coming to the different types of DNA, that is A, B and Z, we will be seeing in detail because they are still found in a respectable amount. So they need to be given a little importance to learn where they are present and what are their functions. So coming to the first one, aDNA. So to observe the structure, what technique is used? X-ray diffraction technique is used, and the dehydrated fibers of the DNA, when they were seen through these X-ray diffraction studies, what were the various points which were identified? That A will exist in a condition where there is low water content and high salt concentration. The existence of A is in high salt concentration and low water concentration. Then it is right-handed, as we have seen in our previous slide, that it is right-handed just like bDNA. Now the bDNA, which is the most prevalent amongst all of the DNA, and it is the major DNA form in which our DNA exists. It is right-handed, double helical. Then the conditions in which it exists is high water and low salt concentration, and it is highly flexible. That we'll be seeing how they are bonded to each other, how it is bound and how it is stored in a very compact manner. So bDNA, it is the most prevalent one in our body. It is right-handed double helical and it exists in high water concentration, low salt concentration, and the most important thing about it is highly flexible. That's why at various stages of cell cycle, you can observe the chromosomes in different forms. Right? Now, the distance we have seen that one base pair and another base pair, what is the height of the rise? So the height of rise in the two base pairs is 3.4 Armstrong and the width is 220 Armstrong. And when we are talking in terms of nanometers, so it will be 2 nanometer, and when we are talking about the distance of one complete helical turn, it will be 3.4 nanometer. Right. One complete helical turn, when it completes one complete circle. And how many nucleotide base pairs did we discuss in previous slide? 10 base pairs are present in a complete helical structure. One helical structure, 10 base pairs. Now coming to the zDNA. zDNA, why does it acquire its name different from the other DNAs? A, B, C, D and E. Why? Because it is in the form of zigzag. It is zigzag in nature, like this. And another important thing is it is left-handed in nature. Unlike the previous ones, they were right-handed, and these, they are left-handed. So where does this Z chromosome actually, or the Z structure of DNA actually exist? When we are saying the most prevalent one is bDNA. So where does this Z exist? Where does this A exist? This is the most important thing. So this Z coexists with B, and where usually the ends of the chromosome, that is the 5' ends, the type of bounding is in the form of Z. The bounding is in the form of Z here. 5' end of the chromosome, they are in the form of Z, zigzag form. Then what is the difference? First one we have seen, it is left-handed. What are the other differences? They are longer, they are thinner than as compared to bDNA. They'll be thinner and they'll be longer. Then how many base pairs are there in one turn? There will be 12 base pairs in a single base pair turn. And for bDNA, it was 10. So this is the difference. And what is this structure? When we are talking about bDNA, we are not talking about any kind of specific sequence. We are just saying that it is right-handed in a one complete helical turn. There are 10 base pairs. But when we are saying about zDNA, how it is made up of? It is made up of altering purine and pyrimidine bases. One purine, one pyrimidine, one purine, one pyrimidine, like that the sequences. So that's why it gives it a zigzag structure. Then, is it a stable form? Is it a stable form of DNA? When we are saying B, when whole of the genetic material, it exists in the form of B structure, then definitely it's an stabilized one. But when we are talking about Z form, so is it a stabilized form? Methylation of guanine and cytosine should occur for stabilization of zDNA. This point you should remember, that for stabilization, what it needs methylation. And one of the things that we saw, these grooves here, the zigzag form. So these are the places where the methyl groups will rest. Otherwise, they'll cause hindrance. So where are these places where these methyl groups actually rest? Maybe the outward or maybe the inward. So that way they try to stabilize the structure. When the sequences, they are not in the form of altering purine and pyrimidine bases, in that case also methylation will make them stable. So methylation is the stabilizing reaction for zDNA. It makes them more stable. zDNA, what is its function? It influences the expression of the gene and regulation. When a gene needs to be expressed, when it needs to be regulated, that is determined by zDNA. So there are many points which should be remembered for zDNA. What are they? They are seen at the 5' end of the chromosome. Altering purine and pyrimidine bases are there. Then methylation of guanine and cytosine residue for stabilization is very important point. And function. So zDNA, what does it do? It influences the expression and regulation of the genetic material. Fine. Now, the qualities of genetic material. So first of it is denaturation of DNA. Denaturation of DNA is also called as melting of DNA. Why the word melting is used and what is denaturation? Whenever a biomolecule, it loses its activity, it is called as denaturation. And here we are using the term melting. So this melting is suggestion that there is something related to water. So the genetic material, the two phosphodiester backbones, they are linked to each other with the help of hydrogen bonds. So when these hydrogen bonds are broken down, they are called as melting. So that's why denaturation of DNA is also called as melting of DNA. Right. So the process by which two antiparallel strands they separate is called as denaturation or melting of DNA. Then what are the features? What happens in this melting? Actually, so what happens is hydrogen bonds are broken down. Phosphodiester bonds are not broken, like here in this structure we saw that the hydrogen bond is broken down, but the phosphodiester backbone, it remains as it is. Primary structure is not altered. This is the primary structure. And when they combine, they join, they form secondary structure. And when these secondary structure, they are superbound on each other, they form tertiary structure. So tertiary structure and secondary structure are broken down. But primary structure, the base sequences, which base will be present in the sequence will not be changed by the denaturation of DNA. Viscosity decreases definitely when we are saying that there is something related to melting. So viscosity will go down whenever there are hydrogen bonds in certain things and they are heated, the viscosity goes down, the thickness goes down. Then UV light. What will happen with the UV light? We have seen that genetic material, it absorbs UV light in the range of 260 nanometer. So what it will do? It will increase the optical absorbance and it will increase the hyperchromicity. That is, now it will absorb more light. The OD will be less. Optical density will be less. It will be absorbing more light. So these are the features. Hydrogen bonds are broken down. Phosphodiester bonds are not broken down. They remain as it is. Thus, the primary structure is preserved. Only secondary and tertiary structure is broken down. Viscosity is decreased. Then optical absorbance, it increases. It starts increases more amount of light, because now purine and pyrimidine bases, they are available for absorbing lots of ultraviolet radiations. Then what is melting temperature? So the midpoint at which the strands separate is called as melting temperature. It is the midpoint of the temperature where the melting occurs. It's called as melting temperature. Now, what are the factors which will be influencing this melting temperature? So, the first thing is GC pair. GC, they are bonded together by triple bond, and AT, they are bonded by double bond. So, it is quite easy to break the double bond as compared to the triple bonds. Okay. GC will make the melting point higher, and higher amount of AT will make the melting point lower. Salt concentration, when we are seeing the salt concentration, if there is an increase in salt concentration, there will be increase in the melting point. 16.6 degrees centigrade will be increase in the melting point of genetic material. Now, another thing, that is formamide, it destabilizes the hydrogen bond. It is responsible for breaking the hydrogen bond. Thus, there will be decrease in the melting temperature. Now, what are the applications of denaturation of DNA? So, increased optical absorbance. Then in recombinant DNA technology, when the genetic material, it needs to be separated, there it is important, rDNA technology. Then in various blotting techniques. So what are the blotting techniques that we'll be seeing? Blotting techniques like northern blotting, southern blotting, and western blotting. These are the major blotting techniques. In these blotting techniques, the strands, they need to be separated, and then they are run and they are baked over a nitrocellulose sheet. So there they need to be separated. Thus, one need to determine the denaturation temperature of the genetic material. There it has its major application. Now, coming to organization of DNA. How they are organized. They cannot be randomly, loosely placed, because in a eukaryotic cell, they cannot lie like that anywhere. They need to be systematically packed at somewhere. So where they are present in the nucleus. When it comes to prokaryotic cell, they are loosely lying anywhere. For prokaryotic cells, they will be loosely lying. And when we are talking about eukaryotic cells, they'll be in a very systematically packed manner in the nucleus. Well organized way in the nucleus. Now what are the level of organization? So first level of organization is DNA double helical structure. Then after that 10 nanometer chromatin fibril. Then 30 nanometer chromatin fibril. Then nuclear scaffold form, that is the interphase chromosome. They will be highly compact. They will be non-condensed loop, and in the metaphase they will be in a highly decondensed form. Now DNA double helical structure. So DNA double helical structure is the first level of organization. That is the first level. They are just wounded on each other. And what are the characteristics? All the characteristics that we learned for the Watson and Crick model, they'll be applied for the DNA double helical. The diameter is 2 nanometer or 20 Armstrong. DNA double helical. Now, what is a 10 nanometer chromatin fibril? The most important thing. So, what is it? It consists of nucleosomes here. Consists of nucleosomes. And these nucleosomes are separated by linker DNA. So, what is a nucleosome? It is a mixture of protein and nucleic acid. Protein and nucleic acid forms nucleosome, and this strand is called as linker DNA. So it consists of nucleosome separated by linker DNA. And what is this nucleosome? Nucleosome is nucleoprotein, nucleic acid and protein. Now, if this is a whole bundle of the protein, that is the histone octamer, the genetic material is wound over it how many times? 1.7 times. It even does not take a complete 2 turns. It takes 1.75 turns over the histone octamer, and it is left-handed helical disc. It is not like right-handed, it is left-handed helical disc, and the genetic material is wound at how many turns? 1.75 times it is wound over this protein. Now, the linker, this linker, what is the length of this linker? It is 30 base pairs. And what is the length of this 1.75 turn? It is around 145 to 150 base pair. 145 to 150 base pair. This is your protein. This protein, histone octamer, is made up of 8 protein molecules which belong to 4 different categories. It's an octamer, that is 8 individual protein molecules are there, and these 8 are belonging to 4 different groups. This linker, let us draw one more diagram. This linker, even this linker is not alone. It is bound to another protein, that is H1 protein. It is a monomer, it is single, and it is bound with the 30 base pair hanging arm. So, how it will look like? This is a nucleosome, this is a linker, this is a nucleosome, this is a linker, nucleosome, linker, nucleosome, and linker. So, what it is giving a look like? Beads on a spring appearance. When they are seen through electron microscopy, they'll appear like beads on a string appearance. So, they are left-handedly bound on the protein. Then there is a linker segment which is 30 base pair long, and it gives a look like a bead on a spring appearance when it is seen through electron microscope. So this is about 10 nanometer chromatin fibril. I hope we are understanding till now what we have learned. This is the diagram, how it will actually look like. So here you can see H3 and H4, H2A and H2B. They are present in 2 number. Each and every is in dimeric form. So that's why it is forming a octamer, histone octamer. And DNA double helix is bound on this histone octamer. Here, stone H1, which is bound with the linker. This is indication of linker. And this turn is 1.75 turns. And how many base pairs are there in this? Approximately 145. And what is the length of linker DNA? That is 30. Here the histone H1 is in the monomeric form. And here they are present in the dimeric form. Each and every histone protein is in the double. This we have seen very well, that it is left-handed, 1.75 turn, and the diameter of nucleosome is 10 nanometer. That's why it is also called as 10 nanometer fibril. Now, coming to the histone protein. Histone protein. So, histone protein is the most abundant chromatin protein present in the genetic material. Then, what kind of protein it is? It is basic in nature. Genetic material is acidic in nature. Genetic material is acidic and this protein is basic. So, they are bound to each other very well. Acidic and basic. So, it is very easy that the proteins, if they are bound to the genetic material, definitely it will be basic. Then, the carboxyl terminal. The protein are made up of 2 terminal, N dash and C dash. N is for amino terminal. Amino terminal means it will have a free NH2 group, and C terminal means it will have a free COOH group. So, the carboxyl terminal is 2/3 hydrophobic, and while the amino terminal is 1/3 rich in the basic amino acids, that is arginine and lysine. The 2 amino acids which are richly present at N terminal is arginine and lysine. Okay. Then these histone proteins, when they are formed, they are not directly used for nucleosome formation. They have to undergo 6 different types of post-translational modification. 6 types of post-translational modification before actually being used as a bounding material for the genetic material. Right. So, we have seen how many different types of histones till now? We have seen 5 types of histones. So, what are these 5 types? H1, H2A, H2B, H3 and H4. These are the 5 types of the histone proteins. Amongst these 5, they are given names. So, what are the names given to them? The core histones and linker histones. So, let us see what are core histones and what are linker histones. Core histones are responsible for forming nucleosome, the bead material that we saw. This is the core histone and it is made up of H2A, H2B, H3 and H4. They form histone octamer. H3 and H4 forms tetramer and H2A and H2B forms dimers. Right. H2A and H2B, they are rich in arginine, and H3 and H4, they are rich in lysine. So, they are basically basic in nature. When we are saying H2A and H2B, it will be very easy for you to remember the A for H2A and A for arginine. And when we are saying H3 and H4, just remember the basic and you can put the basic amino acid lysine over here. So H2A and H2B. H2A, A stands for arginine, and H3 and H4, they stand for lysine rich regions. Got it? Linker is H1. Linker is H1. It is loosely bound to the nucleosome in the linker region, which is 30 nanometer. Now, non-histone proteins. There are various proteins which are involved in further genetic material regulation, replication. So, they are called as non-histone proteins. So, where does they participate? DNA replication, DNA repair, RNA synthesis, RNA processing, and histone protein needs to be bound with the genetic material. So, they are basic in nature. But non-histones, they are mostly acidic in nature because they don't need to bind. They just need to react. So, they are mostly acidic. And histone proteins, they are used for making the genetic material compact. So, they are very small nucleoproteins. Other non-histone proteins, they are larger because they are enzymes. They have to perform various enzymatic reactions. So, they are larger. So, we have seen 10 nanometer chromatin fibril. Now, we will be seeing 30 nanometer chromatin fibril. So, what is a 30 nanometer chromatin fibril? It's a solenoid. The 10 nanometer fibrils are joined in a compact form. When a 6 nucleosomes, they form a compact form, they are called as 30 nanometer chromatin fibril. And the name that they have got, this 30 nanometer and 10 nanometer, that is because of the thickness. The thickness give its word 30 nanometer chromatin fibril and 10 nanometer chromatin fibril. The another word for 30 nanometer chromatin fibril is solenoid. The word given is solenoid. Now, coming to another characteristic of genetic material, that is euchromatin and heterochromatin. Euchromatin, we biology usually use the word U for true. So, the genetic material which is actually used for giving messages or performing transcription, they are called as euchromatin. They are less densely packed, they are loosely packed because they need to perform transcription and then translation. So, they are loosely packed. Then, when they are loosely packed, they will be taking stain in a very light manner. They will be stained lightly. Then another is heterochromatin. Heterochromatin is very, very densely packed. Why is it densely packed? Because it does not require to perform transcription. It is transcriptionally inactive. And when it is very, very dense, it will absorb lots of stain. So, it will give a very dense stain. Chromatin will give dense stain when it is in the form of heterochromatin, And when it is in the form of euchromatin, it will absorb very little stain, it will be lighter, and it will be transcriptionally active, it will be loosely present. Heterochromatin, again it is of two types, constitutive heterochromatin and facultative heterochromatin. So what is this constitutive heterochromatin and facultative? The one which is always inactive. It won't perform any function ever. Means constitutive, always. It is once a heterochromatin, always a heterochromatin. Facultative, it can become transcriptionally active at times. So it is facultative. So where this constitutive is present, constitutive heterochromatin is present in the centromere and the chromosomal ends of the telomere. They are present at two places, centromere and chromosomal end of the telomere. And facultative, it can be widely distributed in the genome. It won't be present at a particular point because it can become active at some point of time. Thus, constitutive heterochromatin and facultative heterochromatin. Now, supercoiling of DNA. DNA can be present in a relaxed or superhelical structure. The bDNA is usually relaxed. We have seen at the very starting that it is the flexible one. Why flexible one? Because it cannot be rigid. It needs to perform replication and transcription. So it is flexible. And the B form is thermodynamically mostly favored. Why? Because it needs to perform replication, repair, various other things. Now, the supercoiling. The supercoils can be positive supercoil or negative supercoil. What does this supercoil actually does? So, again, it is a form of packaging of DNA into compact structures so that it does not get mingled, it does not get tangled with each other, and it does not get distorted. That's why it needs to be properly packed. So supercoil again performs the function of proper folding of genetic material. Another function of supercoil is it facilitates the separation of the DNA strands. When the DNA strands, they are not formed in a proper way, they need to be repaired or they need to perform recombination and meiosis. These supercoils, they serve very important function. Function of supercoil can be seen in repair and recombination. Repair and recombination of DNA. Now, let us talk about an important enzyme, that is topoisomerase. So very simply, what is the function of topoisomerase? It is a nicking enzyme. It causes break in the genetic material. So why these breaks it needs to perform? For relaxing or inserting the supercoil. Topoisomerase is responsible of forming a nick, forming a small break for releasing the genetic material. And why it is doing this? So that it can insert the supercoil. The supercoil can be relaxed or it can be inserted. Now topoisomerase, it is of two types, type 1 and type 2. Type 2, it makes single stranded break in a negatively supercoiled DNA double helix. Phosphodiester bond is interrupted. When we were seeing melting temperature, we saw that the hydrogen bonds are broken down. Here, which bond is broken down? Phosphodiester bond is broken down. Topoisomerase, it will remain bound to the phosphoryl group at the incision site by covalent bond. And till when it will be attached to that phosphoryl group? Till the nick is not released. And it is non-energy dependent reaction. It is a passive reaction. Then the two classes of topoisomerase, one is type 1A and type 1B. So what is the difference between type 1A and type 1B? The type 1A tyrosine residues of the topoisomerase is bound to the 5' phosphoryl group of the nucleotide, and in type 2 the tyrosine residue of the topoisomerase will be bound with the 3' phosphoryl. In both of the cases, remember, tyrosine is the amino acid which will be bound to the phosphoryl group, but you have to remember that in type 1A it is bound to the 5' end, and in type B it is bound to the 3' end. Just that. Type 2, dimeric enzyme that bind to the double stranded DNA, and it causes break in both of these strands. So very easy to remember, topoisomerase 1, it causes break in one strand, and topoisomerase 2, it causes break in both of these strands. So when a single stick we need to break, it is quite easy for us to break. But if we are holding two sticks together and we need to break it, we need to apply more energy, more strength is needed. So when two strands are broken down, it is energy dependent. Very easily you can remember, if it is topoisomerase type 2, it means two strands are now broken down, so energy would be needed. And in topoisomerase type 1, only one strand needs to be broken down, and thus it is non-energy dependent step. Don't forget, topoisomerase type 1 and type B. Which amino acid of them is bound to the phosphoryl group? Tyrosine. Tyrosine is bound to the phosphoryl group. For type 1A, it is bound with the 5' phosphoryl, and for type 1B, it will be bound to the 3' phosphoryl group of the nucleotide. Now, these topoisomerases are very important point for creating the drugs. If bacterial topoisomerase are inhibited, it will further make the reaction of replication quite complex. So remember the name of the bacterial topoisomerase inhibitors. One of them is Novobiocin, Fluoroquinolones, Nalidixic acid. Remember the names. And in human topoisomerase inhibitors, they are very important for chemotherapy. So the one which are used for inhibiting the type 1 topoisomerase, they are Irinotecan and Topotecan. And the one that is used for inhibiting the type 2 inhibitors, type 2 topoisomerase, there are four types, that is etoposide, erythromycin, tonorubicin and idarubicin. Don't forget to remember the names. The inhibitors of topoisomerase, they are an important point in chemotherapy. Now, central dogma. What is the central dogma of molecular biology? DNA can replicate, that is, it can make DNA to DNA. Then it can make RNA, DNA can make RNA, and RNA can make protein. DNA to DNA, DNA to RNA, and RNA to protein. And this process is called as central dogma of molecular biology. DNA, it replicates and forms DNA, RNA, and then DNA transcripts and forms RNA, and RNA, it translates into protein. And this is how the genetic material, it gives instructions for further reactions. Now, with the advent of Human Genome Project, there are three terms which one needs to remember. What is a genome? A complete set of genes of an organism is called as genome. What is a transcriptome? The all type of RNAs which are formed by the genome of an organism, they are called as transcriptome, and the complete type of proteins which are formed from these RNA, they are called as proteome. So now this era, it is an era of proteomics. That is, we have understood what is our genome, we have identified the genetic material, we have come across the base sequence of all of the genetic material through sequencing methods. Now we need to study the complete proteins, the faulty proteins after they are being formed, where they are not properly being utilized or not they are modified. So this is an era of proteomics. We are living in era of proteomics. Now, how many number of base pairs are there in our genome? So 3 million base pairs. And how many exons are there? So what is this exon and intron? Let us understand these two terms also, exon and intron. The euchromatin that we were seeing and heterochromatin, they are present in the genetic material. But when we are talking about RNA, when RNA is formed, RNA is itself made up of exon and intron. Exon can form proteins and introns, they cannot make proteins. So when an RNA is formed, it will be made up of small parts, intron, exon, intron, exon, or any sequence can be like that, exon, exon, exon and intron, depending on the type of protein. So before making it translationally active, a process is done, that is splicing, and these introns are removed. So what is the total percentage of exons in human genome? So it is approximately just 1.5 to 2%. That is quite less. We are utilizing very small part of our genome. So the number of protein coding genes in human genome is approximately 20,000. Approximately it can be little more than 20, or can say it can be between 20 to 30,000. And how much amount does this 20 to 30,000 genes contribute in DNA? Just 10 or 15% of total DNA. So this was it about all theory. Let us just see what types of questions are being asked on this topic. So question can be, which of the following is false? So one can ask that what is the ratio of A is to T, and which of the following is false, sorry. So first of the option is ratio of A is to T and G is to C is approximately equal to 1 is to 1. Ratio of A is to G and T is to C is approximately equal to 1 is to 1. A plus T is equal to G plus C. A plus C is equal to G plus T, and A plus G is equal to C plus T. So what would be the answer? Seeing the question and knowing the theory, this question is based on Chargaff's rule. It is based on Chargaff's rule, and what we have learned in Chargaff's rule is the number of purine will be equal to the number of pyrimidine. So first one, ratio of A is to T and G is to C is approximately equal to 1 is to 1. So this is not possible, right? Because A pairs with T. Now coming to the option B, ratio of A is to G and T is to C is approximately equal to 1 is to 1. So yes, ratio of A is to G and T is to C is approximately equal to 1 because it satisfies our Chargaff's rule. Now option C, A plus T is equal to G plus C. So yes, this option also satisfies our Chargaff's rule. A plus C is equal to G plus T. This also satisfies our Chargaff's rule. A plus G is equal to C plus T. It does not satisfy our Chargaff's rule. So the answer would be B, C and T. Now, what is true about DNA structure? Purines are adenine and guanine. Pyrimidines are uracil and cytosine. So, in pyrimidine, they have written uracil instead of thymine. And in DNA, what is present? Thymine is present. Watson and Crick discovered structure in 1973. So, deoxyphosphate backbone with base stacked inside. So yes, bases they are stacked inside. This is true. And the year in which it was discovered, it was in 1973. If you can remember the very first slide, we have written over there that it was discovered in which year? It wasn't discovered in 1973. It was discovered in 1953. Watson and Crick discovered this structure in 1953. So, the option third is right till now. The A and B, they were wrong. Mainly consists of left-handed helix. No, it consists of right-handed helix, that is in the form of bDNA. And the last one. So, this is also wrong. Last one, 5' to 3' phosphodiester bonding is present. Phosphodiester bond is present, but the bonds they are present in 3' to 5' direction. When we are talking about the sequence of DNA, they are read in 5' to 3' direction. But the bond is always read in 3' to 5' direction. This we have seen in our chemistry and metabolism of nucleic acids. There we have seen very clearly that what is the writing pattern of the genetic sequence and what is the writing pattern of the bonds. So it is from 3' to 5'. So again the last option is wrong. So the answer is C. Now if a sample of DNA, adenine is 23%, what will be the amount of guanine? Adenine pairs with thymine. So adenine plus thymine, how much it will make? It will make 46. So what is the remaining amount? What is the remaining amount? That is the 54. If 54 is the remaining amount and it is asking about guanine, we'll just divide it by 2. We'll be getting 27. So the amount of guanine is 27% because adenine paired with thymine and we got 46 and the remaining is guanine plus cytosine. So this 54 is being contributed by guanine and cytosine. Individually, they are 27%. So the answer would be D, 27%. Now, which is a true statement about DNA structure? All nucleotides are involved in linkage. They are anti-parallel. They are parallel. Bases are perpendicular to DNA attached by hydrogen bonds. So, the only thing that is untrue over here is parallel. They are not parallel. They are anti-parallel. That is, one is from 5' to 3'. The another one is from 3' to 5'. And all the nucleotides, they are involved in linkage. No one is lying free like that because that is making the backbone. Then bases are perpendicular. Yes, the bases are perpendicular to the structure. And between them, there are hydrogen bonds that we have seen very well. Now, the two strands of DNA are held together by, so the bonds which is present between the two strands of DNA, the nucleotides, they are bonded with each other with the help of hydrogen bond. So the answer would be hydrogen bond. Which form of DNA is predominantly seen? We have seen clearly from the starting of the lecture that it is the B form, that is the right-handed helical. A, C and B, they are right-handed. Z, it is left-handed because it is out of the category. So you can easily remember it is left-handed. And where this Z is present, it is present at the ends of the chromosome. So the answer is C. The most predominant form of DNA is bDNA. Now what is Chargaff's rule? Chargraph's rule, we have seen the amount of purine is equal to amount of pyrimidine. So the answer is A. A plus G is equal to T plus C. Now, a nucleic acid was analyzed and found to contain 32% adenine, 18% guanine, 17% cytosine and 33% thymine. The nucleic acid must be. So, the amount of adenine is 32 and adenine pairs with thymine. So, just quickly go to the amount of thymine, and amount of thymine you can see over here, it is 33%. Fine, okay, 32 and 33 is quite near. Then go for looking for guanine. Guanine, they are showing 18% and cytosine, they are showing 17%. So that is also quite near. So this is giving us the full condition of double stranded DNA, because the amount of adenine is equal to thymine and amount of guanine is